Okay, I think we have to begin now. Today's topic is about histology of muscle tissue. Muscle is specializing for contraction, causing movements and locomotion. As you know, there are three types of muscle tissue, which include the skeletal muscle, a widespread type of muscle we have connected to our skeletal system. But we also have muscles of the heart, cardiac muscle, and muscles of visceral organs, smooth muscle. Skeletal and cardiac muscles are striated because we can see striations under light microscopy because of arrangement of their contractile elements in a banding type of manner. And these are striated as opposed to the smooth, which is not revealing such striations. Skeletal muscle is voluntary, whereas cardiac and the smooth muscles are involuntary. The widespread muscle type we have to begin with is the skeletal muscle, which, as I said, is largely connected to the skeletal system of our body for locomotion. But we have a small number of such skeletal muscles structurally identical but not connected to the bones, like those found in the tongue, esophagus, and a few related regions. The muscles are not connected to bone. When we look at organization of skeletal muscle as a whole, as an organ, is wrapped by dense, irregular type of connective tissue. This connective tissue is named as epimesia. The organization is like nervous tissue. Then epimesium sends trabeculae dividing fascicles or bundles. Such fascicles are wrapped by perimesium. Then individual muscle cells, which are elongated and as a result named as muscle fibers, are covered by endomesia. So that's how our muscles are organized. When we look at an individual muscle cell, which look like this, which is covered by endomesium, is about 10 to 100 micrometer in its diameter and could measure several centimeters. It is made by fusion of many myoblasts, so it is multinucleated. Such nuclei are located peripherally and they are following the shape of the muscle cell, so they are also rod-shaped, elongated. It has the basic organelles, some of which are relatively well formed, and one of such well formed structures are the contractile elements, the myofibrils, along with which we have to have plenty of mitochondria, glycogen, and others. Endoplasmic reticulum are well formed. The muscle cell is packed by elongated, parallel running contractile myoproteins named as myofibrils. Here you can see the cell protoplasm, which we name sarcoplasm. The cell membrane is named as sarcolemma. So the sarcoplasm is full of myofibrils. Outside of the cell, tightly applied to it, we have quiescent stem cells, and these stem cells are named as satellite cells. They represent up to about 2 to 7% of the nuclei, and they are important stem cells in case these skeletal muscle cells need to multiply, regenerate. They are the source. When we look at the myofibrils are about 1 to 2 micrometer in width, and they are long end-to-end chain-like arranged contractile proteins, making altogether about 2/3 of the dry weight of the muscle itself. So we have to see them now. When we look at them, they are specifically organized so that they are the reason for the striations of skeletal muscle, which make it appear dark stained region and light stained region. So we have dark light region of the muscle cell under light microscopy. The light bands, the light regions, are named as isotropic band because of their formation mainly from identical related type of proteins, which under polarized microscopy, they are monorefulgent. They don't change polarized light. So because of this, they are named as isotropic. And this I band is bisected by a region named as Z disk. So here we have an I band divided by Z disk. Here we have another one divided by Z disk. In between we have the A band. So that's what the I bands are. But at intervals, we have partly the light region overlapping a thicker, darker region. Because this region is made by two types of fibers, it is changing polarized light, so it is not identical. It is not isotropic, but it is an isotropic band. So because of this, it is named as A band. It is a region overlapped by thin filaments over the thick filaments. The thin ones are coming from either ends. Here you can see the reddish outline divided by the green. Green is the Z disk. The reddish, the pinkish ones are the I band region. The blue one is the A, the dark one. So the dark one is overlapped by the I band region. So this region is an isotropic band. There is a region in between where the I band region, the thin ones are not reaching, is named as H band. Right in the center of H band, there is special protein cross-linking the thick filament, the blue ones. So that midline region is named as M line. So you see A band is also divided by H band. H band is cut by M line. So that's what is making the skeletal muscle striated. The dark ones, A band, the light ones, I band. Now from a region to one Z disk to the next Z disk is a repeating functional unit of the skeletal muscle. And this is named as sarcomer. So here we have a sarcomer spanning from one Z disk to the next Z disk. It is a region that slides over past each other, causing contraction. It is about 3 micrometer in its length, and it is a functional unit of muscle. Like here, you can see how this animated picture shows you contraction is effected. Look at it. Look at how contraction is taking place. Look at the different bands and lines changing. Some are not changing. Look at when contraction takes place, sarcomeres come nearer to each other. A bands do not change. H band almost disappears. So during muscle contraction, we have such changes taking place, and that's what causes contraction and relaxation. And for this, the myofilaments are mainly 4 types. It is the 4 types of myofilaments that have caused the type of descriptions, structures I've told you. This could be divided into 2. One type are thick myofilaments. The thick one is made by myosin filament. A myosin filament, which looks like this, made by about 300 myosin myofilaments. And this myosin filament, which is made by about 200 myosin molecule, has the following type of structure. Each molecule has a region biochemically, heavy chain, and a paired light chain. The heavy chain itself, excuse me, has 2 regions, a rod-like light meromyosin, and a hinge beyond which it bends with a head named as heavy meromyosin. The light meromyosins are wrapping each other so that for proper assembly, they will get assembled. The light chains are in 2, and they are associating with the region of the head of the myosin where ATP is to be bound. So when this myosin molecule is to assemble and make myosin filament, as I told you, about 200, 300 of this are to wrap and get assembled together. When they are to assemble, here is one myosin molecule with 2 ends, head region, tail region. And these are to be laid down to either direction. Like this one, I'm speaking about how this myosin filament is to be formed from myosin molecule. When this myosin molecule is to form myosin filament, the head, this one, is disposed to either direction, some to the right, some to the left, in an overlapping type, a staggering type of manner, so that you are going to have the head disposed at a different level, because it is to the head region that the actin and ATP are to be bound. It is association of this myosin head with its receptor over actin that would make the muscle slide past each other. So for this, we need to have the head to be located at a different location. So this tail, if it begins here, the next one begins a little bit ahead. The next one a little bit ahead. The next one a little bit ahead. We call this staggering type of arrangement to the either direction, so that you have head on both directions, so the actins are able to slide from both directions. So this is how myosin filaments are made from myosin molecules. So we have one filament formed now, and that filament is a thick, which is going to associate with the thin filament out of the 4 type of basic myofilaments I told you. Then the rest 3, the large majority of myofilaments I told you are 4 types, but they are not limited to 4. I'm going to mention also a few more additional, but let's finish the 4 type. I have already gone through the thick one, then the thin. The thin ones are mainly made by actin, but we also have tropomyosin and troponin. The thin filament mainly made by actin, and troponin and tropomyosin look like this. How they are assembled is as follows. First, actin filament is to be laid down from globular proteins. Here you have this round outlines are representing the globular actin molecules. They are assembling in a helical twisted paired filamentous type of manner. So out of this round globular protein, here you have F. F is referring to fiber. Fibrous actin molecule has been achieved. On each of these globular protein, you have one receptor for myosin. So on every of this actin, there is one binding receptor site for myosin. So now the actins are indicated by the blue. Here you have the yellow outlines are representing the active site for myosin. But this actin is not alone. It is inserted on Z disc, I've told you, but it is also associated with another proteins. One of such protein is a paired filamentous type of protein named as tropomyosin, which spans over about 7 actin molecules at a distance. And when it is assembled over actin, it masks, it covers the active sites over actin. So its function is to cover and uncover the active site on actin. So here when it is assembled, actin is no free. It is the active site is buried. So it cannot associate with myosin. So that's how it is found. But we have a third protein, which is named as troponin, with 3 subunits. One subunit is for bounding over tropomyosin. Like this, here is tropomyosin subunit, which has made it attached to tropomyosin. Then we have a second subunit, which is bounding calcium ions. When there is calcium ions coming, which is needed for muscle contraction, and bound to the second subunit of troponin, the active site, which was covered by tropomyosin over the actin, would be uncovered. So that's why we need calcium ions for muscle contraction. When there is no calcium ion, tropomyosin would not be uncovered. But when calcium ion is available, tropomyosin would show conformational changes and it would be uncovered. So you see, now the active site of actin are all free. It is not like this one. When it is with no calcium, the active site is buried. But when this whitish outline representing calcium are available, the tropomyosin is lifted. So actin can associate with the myosin. So that's the second subunit, what's happening. Okay, so that's how the second subunit works. Then we have a third subunit, which limits the association, which finish when there is no calcium. It inhibits, so we name it inhibitory site of troponin. So troponin is playing role in unmasking and letting mask the active site over actin. So these are the 3 components of the tin myofilaments. So we have 4 types of proteins together making thick and thin filament, associating to reveal those striation type of arrangements. In order to do so, with the help of additional many more proteins, we have the following type of disposition. Always the distance between thick filament and between thick and thin filaments are limited for specific type of arrangement, so that sliding in and out is effectively achieved. And for this, it always thick filaments Here indicated, the thick filaments are indicated here by the blue outline. You can say this blue or whatever, violet, are surrounded equidistantly by 6 thin filaments. The thin filaments are the 3 proteins I told you assembled together. They look like this one. So these ones are surrounding from both sides this thick filament in an equidistance, separated. The thick filaments are separated from each other by about 40 nanometer, and from thin filament by about 20 nanometer. So this type of assembly is what is making the myofibrils. And all this type of attachments are done because of interlinking by additional proteins. If you cut diagrammatically as is represented here, you can see the cross-linkings of such a fibrous type of elements. And such proteins include the following, the basic ones. We have one type of protein named as chitin. This one's the red one, which looks like a spring. It extends from half of each thick filament. This is thick filament here. This is thick filament. These ones are thin filaments. This is Z disc. Here is Z disc. This diagram is showing from one Z to the other Z. It is a sarcomer. So here you can see chitin is extending from a region of M line, from half of thick filament to Z disc. So the thick filament is also anchored over Z disc. There is a second one which is named as nebulin. Nebulins are wrapping a sphere around the thin filament, binding this thin filament again to Z disc. We have another ones named as myomesins. Myomesins are making cross-linking of the thick filament right in the middle. It is the one we said make M line. So the thick filaments are bound together in a special arrangement by myomesin. We have alpha-actinin, which is a component of Z disc anchoring the I bands right in the middle. We have desmin, vimentin, which are attaching the Z disc to neighboring myofibrils. So myofibrils are attached one with another in a special register. We have dystrophin, which is binding actin filament of a myofibril to the cytoplasmic aspect of sarcolemma. This is a plasma lemma. So we have such and many more type of proteins which are linking one component of the myofibrils with the other, so that a special type of arrangement is obtained, and sliding in and out for contraction is very well effected. So that's how the myofibrils are found in skeletal muscle. And then we have additional organelles. I'm going to show you, but if you are following me, I would like to have your comment. Are you with me?
Yes, doctor.
Okay. Then, in order for all this to happen, we need to have energy. We need to have calcium as well. So for this, we have additional organelles. One of such well-formed organelles are sarcoplasmic reticulum. These are the smooth endoplasmic reticula. This diagram is showing part of a skeletal muscle opened. The blue is showing the sarcolemma. The inside brownish ones are the myofibrils I told you. The green ones are elongated mitochondria. Among this, you also need to think there are plenty of glycogen and others. The basic function of sarcoplasmic reticulum is to store and release calcium ion. Now you have seen how calcium ions are needed for muscle contraction. when muscle is to contract, immediately this calcium ions should get out and be available. Then afterwards, they have to be stored in the storehouse. That storehouse is sarcoplasmic reticulum. This sarcoplasmic reticulum in skeletal muscle of mammals are arranged as follows. Each sarcoplasmic reticulum has a tubular regions which anastomose in the middle and terminally gets dilated. We have one sarcoplasmic reticulum for A band. We have another sarcoplasmic reticulum for I band. So that's how they are disposed. So we have A band would be served by one unit. I band is to be served by another unit. So you see the end of the sarcoplasmic reticulum are found as a junction of A and I band. Here is the I band partly, here is the A band, all this is A band. So this is one sarcoplasmic reticulum, and here part of the second one is visible. So here at the end of each sarcoplasmic reticulum, the tubular structure is dilated, and such dilated end of sarcoplasmic reticulum is named as terminal cistern. So I'm telling you, a sarcoplasmic reticulum has a tubular region, sarcotubule, and two terminal cistern. So that's what we have. And a terminal cistern is found as a junction of A band and I band. That's one important thing to know. And the sarcotubules are anastomosing, especially in the region of H band. Here is an H band, and right in the H band region, sarcoplasmic reticulum, sarcotubular part of it is anastomosed. The ends are dilated. It is in the ends that we have the calcium ions. So it is from the ends, which we named as terminal cistern, that calcium ions are sequestered, stored, and be released, and be again stored. Then in order to release this, we have to have muscle impulse propagated and arriving here. The impulse is to come from the plasma membrane, from sarcolemma. And this sarcolemma, which gets its impulse when it is innervated, has to get deep inside as transverse projections running between adjacent terminal cistern as T-tubule. Here is a transverse tubule getting deep inside between adjacent terminal cistern. We name it T-tubule or transverse tubule. So what you see here is one terminal cistern, T-tubule in the middle, and another terminal cistern. These three, which are found as a junction of A-I band, make what are named as triad. So when impulse is generated by innervation, impulse comes through along the plasmalemma, through transverse tubule, to the vicinity of where calcium ions are stored. And then this propagates and makes the calcium ions be released in the vicinity of myofibrils for contraction. So that's how the plasmalemma, sarcoplasmic reticulum are working as a unit. In order to generate the energy needed for ATP production, we have plenty of mitochondria nearby. Glycogen is also available. So that's how the sarcoplasmic reticulum is found. The mitochondria are also abundant. And that's what I can tell you about normal histology of skeletal muscle cell. But, excuse me, there are different types of skeletal muscles. These are divided based on maximum rate of contraction, as fast or slow. In physiology, you see this. Major pathway of ATP synthesis, oxidative phosphorylation, or glycolysis. Based on this and related additional density of capillaries, for example, number of mitochondria, level of glycogen. We have different types of skeletal muscle. Generally, they divide them into three. One group are named as slow oxidative muscle. These are small diameter fibers which are suited for slow contraction, but over a long period of time without fatigue. So they sustain slow contraction. And as a result of additional features such as many mitochondria, many capillaries, much myoglobin, they are dark red in color. They are aerobic, and they have high oxidative enzymes and low phosphorylase. So we have one group of such muscles. Then opposite to this, we have a large diameter muscle fiber, which are rapidly able to contract, but short-lived, very strong, very quickly. And they have few mitochondria, few capillaries, and others. And we have this type of low myoglobin, mitochondria, capillaries are less. We have extreme different type of muscle. And the third ones are in the middle. So a skeletal muscle we have in our body are not identical. Some are for fast, short-lived contraction. Some are for slow, sustained contraction. This differs. Good example is, for example, muscles of our eyelid, muscles of the extraocular muscles, which demand very fast contraction. But we also need to have muscles of the back, which should contract slowly over a longer period of time. So our muscles are different type, but they are mixed up. It is a proportion. So depending on the type of muscle, we have our skeletal muscle makeup is different. So that's what we have as the different type of skeletal muscle. I will leave this for you to read. Here is a table summarizing the three different type of muscles. And for such muscles to able carry on their functions, they have to be innervated. The innervation is motor, transmitter is acetylcholine, cholinergic innervation, but the muscle should also be gauged with regard to extent of it is working. So sensory innervation should also be there. So for this, we have motor and the sensory innervation. The motor is making well-formed the myoneural junction we know as a good example of synapse. And for making the synapse, the proportion also matters. Some muscles need to be closely monitored, meaning one nerve can innervate only one muscle. Each of the muscle cells should be innervated, obviously, and one nerve may be given an assignment to regulate one muscle. When that type of muscle is required to be regulated very efficiently, like again the extraocular muscle, like the muscles of our fingers. But in some other cases, the muscles can be regulated as a bulk. Maybe they don't require to be regulated very carefully. So in this type of cases, one nerve can innervate large number of muscles. So it depends on their importance. And this type of innervation give us what is named as motor unit. A motor unit is one nerve plus all muscle cells it innervates. So it differs, like for example, this one indicated by the blue has its motor unit made by this nerve and these two muscle cells. Whereas this one by the green indicated has one nerve plus this muscle, three muscle cells. So the motor unit varies. It varies from 1 to 200, 300 muscles. So that's how skeletal muscles are regulated by their motor innervation. But the state of their contraction should also be analyzed. The brain has to get how the muscle is in tension. And this has a general sensory innervation as well as special sensory innervation. One of such sensory special innervation gave us what is named as neuromuscular spindle. This neuromuscular or muscle spindle works for proprioception of sensation. Proprioception is positional sensation. The state of it is tension. The state of it is contraction, is gauged. And for this, sampling is effected. Sampling is taken. And out of a muscle, say out of the whole biceps muscle, a small region would be sampled and covered by connective tissue capsule. The whole organ looks like a spindle, barrel shaped. And anything that is found within this capsule is named as intrafusal structure, whereas anything that is found outside is named as extrafusal structures. In this intrafusal, we have a double capsule. Right inside, we have a capsule surrounding intrafusal muscle cells and intrafusal nerve fibers. Then between the capsule and the outer covering, we have a liquid, fluid, so that when there is a slight change, slight pressure, the fluid vibrates and makes the sensation more efficient. And for this, the muscle cells are highly modified and have the following type of nature. One type of muscles have their nuclei crowded like a bag in the center. We name this nuclear bag fiber. A second group have their nuclei arranged like this in a chain. We name them nuclear chain fibers. So they are not disposed peripherally like the extrafusal ordinary skeletal muscle. They instead are modified as nuclear bag and nuclear chain fibers. The nerves are also modified as annulospiral, spirally surrounding the region of the nuclei, as well as terminate like a flower spray. We name them annulospiral and flower spray fibers. And there are also additional nerve fibers such as gamma, motor fibers, so that this type of sensation would enable to detect slight changes in tension that is subjecting the whole muscle. So this information reaches to the central nervous system, and if the muscle is excessively contracting or is contracting under the required level, necessary adjustment is immediately taken. And this is what is named as proprioception in the muscle. This type of special sensation is also found in the tendon. First of all, tendon is just nothing but it is an extension of the endomysium, perimysium, and epimysium that would make the muscle extend and get connected to the bone. So that's what it is. Tendon is regular type of dense connective tissue. And this tendon is connecting the force coming from the muscle contraction to the bone. And the state of tension passing to the bone should also be gauged. And for this, we also have same type of innervation, but less developed, and that is named as the Golgi tendon organ, neurotendinium spindle. Otherwise, if muscle is contracting just uncontrolled and with no uniform level, it can even tear the muscle, tear the tendon, or even break the bone. So it has to be regulated, and as a result, tendon also is regulated with regard to the tension it communicates from the muscle to the bone. So that's what I can tell you briefly about the skeletal muscle. If you have any question, now is the time before we move and look at briefly the remaining muscle in muscle tissue. Do you have any question now? ጥያቄ አላችሁ? Are you with me?
Yes, doctor, we are.
Okay. Okay, I think I have to proceed now. The second group of muscle we have are cardiac. Cardiac are mainly muscle of the heart, but they also extend to the big, large-sized veins, pulmonary and vena cava. But generally, when you hear of cardiac muscle, think of the heart, the middle layer, myocardium. Now I'm emphasizing the basic difference of this cardiac muscle from skeletal muscle. One is that it is inserted into a connective tissue right at the junction of the atria and the ventricle. That connective tissue with fibrocartilage region is named as cardiac skeleton. So cardiac muscle are running as a whole, beginning from the junction, and they go down and they come back. That's how they are organized. And the basic nature of cardiac muscle is again for contraction in a somewhat comparable manner with that of skeletal muscle, but there are also impulse conducting, generating and conducting system, whereby the cardiac muscle, without waiting for innervation, are able to generate impulse and conduct it, as well as there are also secretory cells. So cardiac muscles are behaving like neuron in impulse generating, as well as they are behaving like endocrine gland in being secretory. Otherwise, normally they are contractile. They are smaller in size, 15 to 80 micrometer. So they are much smaller. They are cylindrical. They branch and anastomose. They are largely uninucleated, but at some stages you may find binucleated cardiac muscle cells. These are the basic difference. Their myofibrils are like that of skeletal muscle, but a little bit less developed. No mitosis, they are not able to regenerate. There are no satellite cells for skeletal muscle. We said satellite cells are stored stem cells, but these ones do not have it. They have sarcoplasmic reticulum for same purpose. And they are not fused, but highly interconnected by cell attachment, a special type of cell attachment which makes the intercalated discs in a zigzag type of manner. One cell is tightly connected with another. If we resolve this intercalated disc, which looks like a small transverse line under light microscopy, under electron microscopy, it could be resolved to have a transverse limb and a longitudinal limb. A transverse limb, a longitudinal limb. The transverse limbs which cut across the muscle usually correlates with some of Z disc. Look at this. This is a region of I band, and in that region, it correlates with Z disc. In such transverse limbs, the intercalated disc has zonular adherence, intermediate junction, as well as desmosome. So its purpose is to link two muscle cells together, whereas the longitudinal segment is mainly made by gap junction, so that ions are communicating between adjacent cells. One individual muscle should not behave differently. They are to work in a syncytium type of manner. So one muscle contracting means other muscle also need to contract. So for that, they are interconnected in such a manner. When it comes to the calcium storage system, sarcoplasmic reticulum, the sarcoplasmic reticulum of cardiac muscle is not well organized, unlike that of skeletal muscles. One, there is no terminal cister, but there are sarcotubules. These sarcotubules are not well organized, unlike in skeletal muscle. They don't also discretely divide and run in between myofibrils. This is the skeletal muscle I showed you earlier. Look at the myofibrils are discretely divided, but here they are not as such divided. The sarcoplasmic reticulum are not entangling efficiently like in the skeletal muscle. So they are not as well formed. I told you there is no terminal cister. So otherwise there are well formed T-tubules. The T-tubules are well formed. And they also make association with sarcoplasmic reticulum, but not with terminal cister, because there is no terminal cister. And this index of sarcoplasmic reticulum is found at Z disc. T-tubules are running at Z disc. It is not at the junction of A and I band. So what we have at Z disc is the contact of one transverse tubule and separate sarcotubules. This makes dyad. Triads are three structures of skeletal muscle like this one, but for cardiac muscle, we have dyad. One well-formed T-tubule and transverse tubules. The function is for same purpose. Mitochondria for same purpose are present. And in some of the cardiac muscle, we have such vesicles. Such vesicles are mainly located around the atrial appendages, and they are secretory. They produce some secretory products, and they are making the cardiac muscle also behave like endocrine glandular cells in a sense. They are also impulse generating and conducting system, as I told you. For such impulse conduction, we have the impulses to be generated at the junction of superior vena cava and right atrium at sinoatrial node, internode, atrioventricular node, and in the ventricles we have bundles, atrioventricular bundle, and Purkinje fibers. So the impulse generating and the conducting system, you will study this more in detail in cardiovascular system, is also a function of cardiac muscle cells. So that's what I can tell you about the cardiac muscle cells. Then finally, we have to see a few things about smooth muscle. So I finished about cardiac muscle. ጥያቄ ካላችሁ ጠይቁ። ከዛ to move to smooth muscle cells. Okay, smooth muscles are smooth is not because they do not have myofibrils. It is the arrangement. It is the arrangement, otherwise they also need to have myosin and actin for contraction. So one important thing to note is this. Second is that smooth muscle can be found isolated, but normally they are also making bundles of sheet overlapping. They are spindle in shape, broad in the middle, tapered toward the end, centrally placed oval nucleus. Their overlappment makes them have this type of organization, which makes them use as wrappings. Their size tremendously vary from as little as 20 to as big as 600 micrometer, depending on the organ. They have no T-tubule. They have another system named as caveolae which would let the impulse conduction system propagated towards the inside, otherwise not a tubule. And they also have gap junctions, and some cytoplasmic vesicles are also present. And the way the myofibrils are organized is in a different type of manner. The myofilaments are attached to dense oval bodies, like this one, which have attachment to the interior of sarcoplasm, as well as freely inside the plasma membrane, associated with several proteins. And this type of disposition makes them to be strongly contract. When they contract, they cause this type of foldings, including the nuclei are folded like this when they are relaxed, when they are contracted. So it is the arrangement of the myofibrils that would give rise to a smooth nature. Otherwise, smooth muscles also need to have myofibrils. Otherwise, how could they contract? So it is arrangement, not the presence or absence that matters. So that's what I can tell you briefly about smooth muscle, leaving the others for you to read. I will send you the PowerPoint at the end, as well as you have to enrich the discussion. Then saying this, there are also other cells that are able to contract. This include myoepithelial cells, which are developmentally epithelial, but they are contractile. They are found in some glandular tissues, like here. You have a duct between the duct cell and the basal lamina. Inside, you have such contractile cells. They are able to contract because of, again, the myosin and the actin. So these are other contractile cells which are behaving like muscle. We have the parasites. Parasites are found around capillaries and the postcapillary venules. They are also contractile cells. They are behaving like muscle. They are myofibroblasts. These are fibroblasts which, when needed, are able to contract. So these are also divided in contractile cells. Otherwise, all dividing cells also are able to contract at a given time, at least when they are dividing. The myoid cells of the testis, in the testis, at the bottom of seminiferous tubules, we also have myoid cells, which are contractile. So contractility is not only the function of muscle, but related cells. So you can say today's topic is muscle tissue and the contractile tissue. When we say contractile tissue, we have professional contractile cells, semi-professionals, as well as all dividing cells are also contractile. I think that's all I have for you for this lecture for the day. Thank you for your attention. If you have any question, feel free to ask. Otherwise, I've gone through and have tried to do my best. I'm struggling with this flu pandemic. I'm working from the house. I'm very much tired, but I've tried to do my best. Thank you for your attention. ጥያቄ ካላችሁ ጠይቁ።
እሺ ቻት ቦክስ ላይ ትንሽ እንጠብቅ ዶክተር።
ሶሪ
ቻት ቦክስ ላይ ትንሽ እንጠብቅና
እሺ
እሺ
እሺ So I think I don't see. You can chat also tomorrow. I will have another chance for you. Integumentary system. So you read በቃ ግድ የለም ልሄድ እኔ አሁን እና እልክልሻለሁ እንትኑ።
እሺ ቴንክ ዩ
ኦኬ